Hey, I wanted to hop on here and talk to you a little bit about a gene therapy that was just recently successful in reversing deafness in a child. But I think that this would actually apply across the age spectrum, especially as we get older and we lose some of our hearing, or some people can lose particular aspects of their hearing. So I want to go over this particular article by the BBC called Pioneering Gene Therapy Restores UK Girls' Hearing. So I'm not going to read this for you, it's not story time, but I did want to go over exactly how they did this, how this actually works. And what they found is that this girl, this young child, had hearing loss due to a genetic defect. I think the defect was in a gene called the OTOF gene. So this particular gene impedes the ability for the hair cells for the cells, hair cells that are in the ear to transmit sound. And the reason why is because there's a defect in the ability to undergo exocytosis. So you've got these vesicles here, and usually they're secreted from a region of your cell. It's kind of a cross region between two different areas of your cell called the Golgi apparatus and the endoplasmic reticulum, and they go through what's known as the secretory pathway as opposed to some other pathways that they can go down, and they get packaged into these vesicles, which then fuse with the outer membrane of the cell. And then once they fuse, they release them. Now, I'm assuming that for these hair cells, which I'll show you some images of, that there's some sort of the release mechanism doesn't work somehow, and this OTOF gene is involved in this whole process of releasing or allowing for these hair cells to end up on the outside of the cell, which is where they're supposed to be. So to give you a little bit of background on you, a little bit more background on here, so especially as you are, getting to that age where you're supposed to be talking, that's usually when researchers, or I should say clinicians, identify that there may be some delay in speech development, and that can be largely due to not being able to hear. And because obviously you're getting a lot of feedback and you're learning from the exposure that you have throughout your life. So, especially early on in life. So what they did is they added this therapy, which I'll describe to you in a moment, to her right ear, and then they did a cochlear implant, if I remember correctly, wearing a cochlear implant in the left ear. And then they ended up doing a bunch of repeated tests over time, I think over 6 months, to see if she would regain the ability to hear in her right ear, which is over here, in case you weren't familiar. Okay, so how does this work? What they do, and this is actually something that I've done myself, not on myself, not on other people, in the lab on cells. So I used a variety of different systems. So there are, the systems are called vectors. So if we've done it with what are known as plasmids, which is what I currently do in my PhD, use plasmids to manipulate the genes of cells. But what I did in my master's and some in my undergrad as well was use a virus. And that's actually what they use in this example. So the DNA is added with this harmless virus, supposedly harmless, probably is harmless, and then it's put into the syringe, and the syringe then gets this tube that goes to the cochlea itself. And you can see, this is an example, if you were to zoom into the cochlea, you can see these hair cells aren't correctly linear, they're not correctly placed outside of the cochlea, and you can see that normally it would be more straightforward, and that's because you have this problem with this exocytosis, so they can't release these hair cells appropriately. So what they did is they had this DNA, which is the functional version, presumably, they don't actually go into the details here, but the functional version of this OTOF gene. Because if you have some sort of mutation, that means that, and it can come in any form of mutation. It can be a truncation mutation, which means that the gene itself is, let's say, 60 different read sequences. So it is 60 different DNA pairs, okay? And the mutation cuts off at 40. So it reads 40 and then it doesn't read the remaining 20. And that can be a truncation mutation, or it can be something as simple as what's known as a point mutation, where just one of the DNA base pairs is replaced with something else, which leads to a problem with the reading of that gene, or it gets read, but it doesn't get folded into the correct protein. There's all kinds of different types of mutations. I'm not going to go into all of them, but the point is that the cochlea itself just doesn't work because of this OTOF mutation. Anyway, so they end up injecting, they end up using this DNA, which they put inside of the virus, so the capsid here, and then this DNA held inside the virus is then extremely good at invading into the cell, and they put it into the syringe. So the syringe has this liquid, which is just the suspension liquid with the virus, including the OTOF gene, the actual functional gene, and then they have it travel to this cochlea, and at the hair cells, or at the cells themselves, then they release this virus. The virus then enters into the cell through actually a process that's the exact opposite of exocytosis, called endocytosis, through some form of endocytosis. And then that gene then gets read, and usually it gets read at a level, an abundance that overwhelms the reading of the other gene, the mutated gene. And then in that case, if you have DNA implantation, so the DNA finds itself into the nucleus and it starts getting read, then you start having the production of the correct OTOF protein, which then leads to the correct exocytosis, which then leads to the correction of these hair cells. And now, after 6 months, she was actually basically healed, so that she was able to hear, if I remember correctly. I think I do. Yeah, so just a few weeks later, just a few weeks after this therapy, she could hear again out of that ear. Loud sounds, not like anything soft, but pretty loud sounds. And then after 6 months, she could hear even quiet whispers, which is, I mean, that level of medical technology, that level of ability to fix these kinds of problems, which, I mean, even just like a decade ago or less was impossible. We couldn't do any of that stuff. So it's unbelievable that we can take what nature has provided us. We've learned from nature and used the natural process to then fix a naturally occurring problem in a person. And of course, this opens ethical questions. I'm not going to get into those at the moment. I'm just going to marvel in the amazing feat of being able to turn somebody's life and their future life, especially at such a young age, to turn their life from potentially a disaster, something so difficult to overcome, to having then a potentially completely normal life, just from the therapy using what nature has given us, viruses, and how they infect our cells, and just understanding how viruses infect our cells, and understanding the risk of how viruses infect us and which ones are actually of great risk and whatnot. So just an incredible story, and I just wanted to share. Anyway, till next time.